This is a lecture on recombinant DNA technology. Now, the picture of Narasimha, most of us know, was a god with a lion's head and a human's body. Chimera is another term which is used commonly in the field of medicine, which talks about an animal which was having a head of a lion, the body of a goat, and a tail of a serpent, meaning that different animals' parts were joined together to produce a new structure. When we do the same thing with DNA, that's called recombinant DNA technology. Now, from the exam point of view, what we are interested in knowing is about the restriction enzymes, the enzymes that produce blunt ends and sticky ends. We're also interested in knowing the mechanism of recombinant DNA technology, a word about antibiotic resistance and the blue-white screen, and applications of recombinant DNA technology in the modern world. So there are two types of restriction enzymes. They're called the exonucleases and the endonucleases. Exonucleases are those that cut the DNA from the free ends, DNA or the RNA from the free ends. It could be the 3' end or the 5' end. It starts sequentially from one end and keeps cutting the DNA. The endonucleases, on the other hand, are those that cut within the nucleic acids. So when you're talking about the DNA, the endonucleases can cut the double-stranded DNA either as a straight vertical line producing blunt ends, or can cut in an angle producing overhangings called the sticky ends. Now the blunt ends are called blunt because if we mix these two DNA back again, they will not join to form the original DNA. However, those restriction enzymes that produce sticky ends have areas where there are complementary base pairs and they can anneal and produce back the original DNA. That's why they're called the sticky ends. Now, it's important to know that the sticky ends, the enzymes that produce sticky ends, usually follow the palindrome sequence. There are palindrome sequences in the DNA which is identified by these restriction enzymes, and they cut the DNA to produce these overhangings. These two pieces can rejoin at a later date, and that's why it's called sticky ends. Important to note that whether we are using the DNA of a plasmid, a bacteria, or a human, if we use a particular restriction endonuclease producing sticky ends, the DNA, the site cut will be identical, and you will have a situation where you can fuse human DNA to the plasmid DNA. Now, the technique of recombinant DNA technology, we want to introduce a gene of interest into a bacteria. Why? Because we want to study what is the kind of protein produced by the gene and what is the impact of this protein. Or more often, we are trying to extract the gene product like insulin, like factor 8 for treatment of hemophilia. We want to produce that in large amounts. So we introduce the gene into the bacteria, let it grow in the bacteria, and extract the proteins from the bacteria. This is why recombinant DNA technology is becoming important. For that, what do we require? We require the gene of our interest. We require a vector that can carry the gene of our interest, and we require the bacteria in which we are going to introduce this recombinant DNA. What are the various kinds of vectors? There are plasmids, there are bacteriophages, there is cosmids, there are yeast artificial chromosome, there are bacterial artificial chromosome. The only difference between all these being the amount of DNA that we can pack into these vectors before we can introduce it into the bacteria. So, making the gene construct. We take the gene of our interest and treat it with a restriction enzyme that produces sticky ends. We also take a suitable vector. In this case, we are taking a plasmid. We treat the plasmid with the same restriction enzyme. Now, as I said, if we are using the same restriction enzyme, we get the same sticky ends, and when we make the plasmid and the DNA interact with each other, like this is the DNA, this is the plasmid, we treated it with the same restriction enzymes, they produce the same sticky ends. So when we let both of them join together, there are chances that the DNA of our interest gets inserted into the plasmid, and when this happens, we have produced our recombinant DNA. But there's also another possibility that the plasmid may simply close without taking the foreign DNA. This event is also a possibility. So, after we've tried to create this recombinant DNA technology, now the vector is independent of incapable of independent existence. So we need to introduce it into the bacteria. To do that, we use various treatment modalities called the heat shock treatment or using high concentration calcium. This can push the vector into the bacteria. Once the vector has gone into the bacteria, then we culture the bacteria in a suitable culture medium, usually agarose. And the bacteria will grow to produce colonies like this. How do we know whether the vector of our interest has entered into the bacteria or not? To address this question, we should revisit our plasmid, and our plasmid is such that it has got two sites of antibiotic resistance. Antibiotic resistance for ampicillin, antibiotic resistance for tetracycline. The bacteria that we use is such that it would be sensitive to ampicillin and tetracycline. If the culture medium had ampicillin or tetracycline, the bacteria would die. But if the vector has gone into the bacteria, it would provide it resistance to ampicillin and hence the bacteria will be able to grow. So after making my construct and after trying to insert the construct into the bacteria, if I grow the bacteria in a plate containing ampicillin, only those bacteria which had the resistance to ampicillin would grow. That means that the bacteria which grew had the plasmid of my interest. So this is how we first get the confirmation that the bacteria that we are dealing with has got the plasmid of my interest. But again the question comes that the plasmid, does it really have the DNA of my interest or not? Does it have the recombinant DNA that we were trying to test? Because I said there is a possibility that the plasmid can close and not take the DNA. So how do we address that? To address that issue, we take the help of the second antibiotic resistance, that is tetracycline. The construct of the plasmid is such that the tetracycline antibiotic gene resistance has got the same restriction site that we used when we were trying to treat our primary DNA and tried to make it join to the plasmid. So the restriction enzyme will cut here. Okay. If my target DNA of interest has integrated, it would integrate in between the gene that is conferring resistance to tetracycline, which means that this bacteria will no longer be resistant to tetracycline. It would be sensitive to tetracycline. It would be resistant to ampicillin, but because the resistance to tetracycline has been lost because of my foreign DNA has got inserted in the same place, this bacteria will be sensitive to tetracycline. So what I do is, after I have grown the bacteria in the ampicillin plate, I take an imprint on another medium which has got tetracycline. Those bacteria which do not grow in the medium of tetracycline, those bacteria which do not grow in the medium of tetracycline are those bacteria in which my DNA of interest has joined. Now let's get back to this picture. Here we are using a tetracycline gene resistance. In that my target DNA is being inserted. If my target DNA gets inserted, then this bacteria is no longer resistant to tetracycline. So if the bacteria has grown in ampicillin, that means my plasmid is there. But if the bacteria doesn't grow in the tetracycline medium, it means that my target DNA got inserted. If it grows even in the tetracycline medium, that means that this gene was not disrupted. That means the plasmid is there, but my target DNA is not there. So we compare the two plates. We look for those plates where the we look for those colonies which are present in the ampicillin plate but are not present in the tetracycline plate. These are the 5 colonies that you can see which are present in the ampicillin plate but not present in the tetracycline plate. These are the organisms which contain the DNA of my interest, and these are the organisms which I should subculture for further activity. Obviously there is a problem here. the problem here is that we have to take two imprints, we have to see which colony is not growing, and from there we are supposed to pick it up. Obviously there's a problem. To overcome this, now we have what's called the blue-white screen. Here we create a plasmid with an antibiotic resistance and a gene for galactose metabolism. If the plasmid were present, the bacteria would be able to use galactose in the medium, produce an acid that changes the color of the medium. If not, the medium would be blue color. So we treat the plasmid and the target DNA with the same fragment. We introduce it into the bacteria. Only those bacteria which have got the plasmid will be able to glow because of the antibiotic resistance. In the next step, we see the color of the colony of the organism. If an organism grows in this plate, obviously it was resistant to ampicillin, which means that the vector has gone inside. If the bacteria has the DNA of my interest, it would be in the gene for utilizing galactose, as a result of which the colony produced will be white. In case the vector was present but the target DNA had not gone inside, this bacteria will be able to utilize galactose and produce these blue color colonies. So the white color colonies are the colonies where my target DNA of interest is present, and the blue color colonies are those colonies where the plasmid is present but the target DNA of interest is not present. So by one single culture itself, I can identify which were the bacteria which had my target interest or not. Use of recombinant technology. Recombinant technology is used for studying dangerous viruses like HIV. When a lot of research was being done on that, handling the virus was going to be a problem, so we used recombinant DNA technology. Studying the gene function, producing proteins in large amounts like insulin, factor VIII, production of vaccines, production of monoclonal antibodies both for diagnostic and therapeutic use. Thank you.